Hi all. Today I'm handling Unit 6, Nucleic Acid. So in this particular chapter, you are supposed to learn the composition, functions, classification, isolation, structure and properties of DNA and RNA by synthesis and breakdown of purine and the pyrimidine. So let's see in detail. So what is nucleic acid? Nucleic acid is an important class of macromolecules found in all cells and viruses. So the main function of nucleic acid include the storage and expression of genetic information. Normally, these nucleic acids are composed of nucleotides, which are the monomers made of three components, a 5-carbon sugar, a phosphate group, and a nitrogenous base. The two main classes of nucleic acids are DNA or deoxyribonucleic acid and ribonucleic acid or RNA. So here is the composition of the nucleic acid. In the case of DNA, it is a double-stranded polymer, and the sugar involved in the DNA is deoxyribose. The bases in nitrogenous bases included in the DNA are thymine, cytosine, adenine, and guanine. Now coming to the RNA, it is a single-stranded polymer. The sugar involved is ribose sugar, and the bases, except in the case of thymine, in RNA it is uracil. All other three nitrogen bases are same as in DNA. So this is the composition of the nucleic acid. Now we will see the structure of each part. So this is the sugar part, deoxyribose and ribose sugar part. The structure looks like this. Now this is the phosphoric acid. The structural appearance of the phosphoric acid is like this. Now the nitrogenous bases adenine, guanine, cytosine, thymine, and uracil. Now let's see the functions of DNA and RNA. The function of RNA include it acts as a cofactor in nuclear aerobic synthesis of ATP. It carries biological information. It replicates itself accurately. Now, what are the functions of DNA? In last chapter, we have seen the protein synthesis, how the involvement of RNA in protein synthesis. Okay, so along with protein synthesis, genetic material in viruses, and it initiates the replication of DNA. Now, what are the biological functions of a nucleic acid? It includes protein synthesis, memory, replication of the cell, and of course, genetic information, then mutation. So here are the biological functions of nucleic acid. The very famous Watson and Crick model of DNA. In very lower class also, you might be learned about this DNA model, where the Watson and Crick proposed that the DNA is made up of two strands that are twisted around, which is shown in the picture. So this is twisted around each other to form a right-handed helix called a double helix. And the base pairing takes place between the purine and pyrimidine, namely A pairs with the T. So this is A, the A is paired with the T and C is paired with the G. So in other words, we can say adenine and thymine are complementary base pairs and cytosine and guanine are also a complementary base pairs. So that's all about the Watson and Crick model of DNA. Now we will see the different types of RNA. So messenger RNA, transfer RNA, ribosomal RNA. I'm sure you will be aware about all the three types of RNA because in last chapter, protein metabolism, we have discussed the way different roles carried by each RNA here. So let's see. The first one is the messenger RNA. The messenger RNA is a single-stranded molecule of RNA that correspond to the genetic sequence of a gene, and it is read by a ribosome in the process of synthesizing a protein. Next is tRNA. So this is the structure of the tRNA. Actually, tRNA, it is a small RNA molecule. It is also participate in our protein synthesis, and each tRNA have two important areas, like the trinucleotide region that is called the anticodon, and a region for attaching the specific amino acid. So the structure of tRNA is very important. If it asked in your exam, definitely you must draw this picture. Now next is ribosomal RNA. So what is the function of the tRNA? You might be know. tRNA is to bring amino acids to the ribosome for the protein production. So that is the main function of the tRNA. Now next is rRNA. So it is within the ribosome, the rRNA molecule that direct the catalytic steps of the protein synthesis. That is the stitching together of amino acid to make a protein molecule. So this is also we have seen in the protein synthesis in last chapter. So these are the different types of RNAs. Now we will see the metabolism of nucleic acid. The first one is a purine metabolism. So the purine metabolism, it includes the de novo purine biosynthetic pathway. It consists of three pathways like de novo purine biosynthetic pathway, purine salvage pathway, and the degradation. So the de novo purine biosynthetic pathway, it uses 6 enzymes to catalyze the transformation of a PRPP, that is phosphoribosyl pyrophosphate, into inosine 5 monophosphate or IMP. That is happen by utilizing 10 highly conserved steps. Okay, that is highlighted in the orange color. Then purine salvage. Purine salvage, here the this green line shows the purine salvage steps. So here it recycle the hypoxanthine, inosine, and adenine as a substrate to generate the purine nucleotides. Then inosine and that hypoxanthine that can be further oxidized into xanthine and uric acid in the purine degradation pathway. So that is shown in this blue color. Okay, so this is the overall look of the purine metabolism. So this final product of the purine metabolism, that is IMP, inosine monophosphate, that will split into the AMP and GMP. So the synthesis of AMPs occurs the IMP, that is the final product of the purine metabolism, that combined with aspartate and GTP, forming adenosyl succinate plus GDP and inorganic phosphate. Adenosyl succinate is again split into form AMP plus fumarate. Now the synthesis of GMP is this: when this IMP combined with NAD molecule, it forms XMP plus NADH plus hydrogen ion. And this XMP again combined with the glutamine and the AMP plus pyrophosphate in inorganic pyrophosphate. So this is how the GMP formed. Now we'll see the synthesis of pyrimidine. Here the glutamine plus ATP plus carbon dioxide, it gave a CAP compound. It combined with aspartate and forming CAA. And again it had split into forming two compound, DHOA plus water molecule. And again DHO combined with the energy molecule NAD plus and forming OA plus NADH plus hydrogen ion. And this OA again combined with the PRPP, the first element we have seen in the metabolism of the purine, combined together to forming OMP. OMP is again split into UMP plus carbon dioxide. So this is how the synthesis of pyrimidine occurs. Now about the salvage pathway for the purines and pyrimidines. Actually, the process of recycling the free purines and pyrimidine bases for the reuse in the biosynthesis of nucleotide and nucleic acids. So that is what happening in the salvage pathway. So in the purine salvage pathway, there are two mechanisms. The first mechanism includes the phosphoribosylation of free purines by using PRPP. Here, in this case, two enzymes are involved like HG, PRT and APRT. So here, first the guanine combined with the PRPP to forming the guanine monophosphate, hypoxanthine combined with the PRPP to form IMP, the inosine monophosphate, adenosine combined with the PRPP to forming AMP. So this is the first mechanism. Now the second mechanism involved the adenosine combined with ATP and forming AMP. Now, next is pyrimidine base salvage. So here in pyrimidine, the nitrogenous base uridine and cytidine are converted into UMP and CMP. Uridine and cytosine. Reactions are catalyzed here by the uridine and cytosine kinase. Now we will see the catabolism of purines. So here, the catabolism of purine steps are shown here. The blue color box are showing the involvement of enzymes. Okay, the name of enzymes are highlighted in the blue box. Okay, now the final product of the purine catabolism is uric acid here. Now coming to the catabolism of pyrimidine. So here the first cytosine, in the presence of enzyme cytosine deaminase, one water molecule will convert it to liberate, and water molecule involved, and it liberate as in the form of ammonia and forming uracil or thymine. So in the presence of dihydroxy pyrimidine deoxygenase, it formed dihydropyrimidinase. Okay. So this is the compound from the uracil is a dihydrouracil, and if it is thymine, it forms dihydrothymine. Now another enzyme dihydropyrimidinase, this new compound beta urido propanate and beta urido isobutyrate. It is converted into the another beta alanine for under uracil and beta amino isobutyrate under the thymine. Now we will see the difference between DNA and RNA. DNA, genetic material, it is a genetic material which is present in all organisms and it occurs in nucleus. The quantity is constant here. The sugar involved is deoxyribose sugar and the nitrogenous base in cytosine and thymine. The base pairs are in equal amount here. Two polynucleotide chains are there. It can replicate itself. It controls structure, heredity, evolution, etc. Component of it is a component of a chromosome. The molecular weight include 2 to 6 million. Now coming to RNA, if it is a genetic material of all organism in RNA, it is the genetic material in certain viruses. And here it is nucleus occurs in nucleus. Here RNA is occurs in cytoplasm. The quantity is various here in RNA, but it is constant in the DNA. Now, the sugar involved is ribose sugar. Nitrogenous base include our cytosine and uracil. The base pairs not in equal amount, but in our DNA, it exists in an equal amount. Now, it is a single polynucleotide chain. It is formed by DNA. It involved in the protein synthesis. It is a component of ribosome, and the molecular weight include 25,000 to 2 million. So here is the difference between DNA and RNA. So this question is used to ask in most of our university exam also. So please note it and study well. Okay. Now isolation and purification of nucleic acid. Actually, I'm just brief the whole procedure in 3 basic steps. So you have to write more detail in your assignment. Okay. And we will discuss it more in our seminars also. So the 3 basic steps include rupturing of cell membrane. Next is second step is separation of cell membrane, and last one is purification of nucleic acid. So these are the basic steps involved in the isolation and purification of the nucleic acid. So that's all. Thank you all.